Yes, this one is part 3, which is still we are on chapter 3 on nucleic acid and the replication, metabolism, and the central dogma of this nucleic acid. So let me see the part 3, which starts on the inhibitor of DNA replication. The progress of replication can be stopped by the presence of different inhibitors. This inhibitor may be what? Different drugs, molecules that can be inhibit the replication of the DNA. For example, if you take actinomycin D or dactinomycin, which is an anti-cancer drug, bind the minor groove of the DNA and prevent both replication as well as transcription. Not only this, different topoisomerase inhibitors such as etoposide are used as an anti-cancer chemotherapy, and they inhibit what the DNA replication as well as transcription. So inhibitors of prokaryotic DNA gyrase are used as an antibacterial agent. For example, quinolines, quinoline, which have different classes of drug. For example, ciprofloxacillin from the class of quinoline can be inhibitors of the DNA gyrase, especially DNA gyrase B can be inhibited by this ciprofloxacillin. Nucleoside analog like 2,3-dihydroxyribonucleoside with modified sugar are a competitive inhibitor of DNA polymerase, which block a DNA chain elongation. And the different anti-HIV drug like ART drug like azidotiamidine can also be inhibit this chain elongation drug of DNA. So there is the concept of mutation and the DNA repair here, and a very important concept for health student, because knowing the concept of mutation and the DNA repair is very critical for to know what the mechanism of action of different drug in our body. So this DNA must be faithfully replicated, but sometimes mistake can be occurred. The mistake occurred in DNA repair can lead us different disease, maybe different genetically inherited disease or other disease. So E. coli take less than 1 hour to copy 5 million base pairs in its single chromosomes, and DNA polymerase can have, as we mentioned before, the proofread and the correcting error function, repair mismatched base, remove abnormal bases, and reduce an error rate from 1 in 10,000 to that of 1 in 100 million bases. So these DNA polymerase have the proofreading function in prokaryotic cell. In the same fashion, DNA polymerase delta can have the proofread and the correcting error function in case of eukaryotic cells. The human cell copies at it is 6 billion base and divided into daughter cells in only few hours, remarkably very accurate way, and only 1 error per 100 million base can be occurred, and associated error with cell cycle can be almost 13 errors per cell cycle can be occurred. So despite the proofreading and the mismatch repair during replication, some mismatched base do persist and maybe but continue, or hidden error may be associated with this DNA repair, and the additional problem may arise from this DNA damaged by mutagens and the chemicals produced in a cell inhaled or absorbed from the environment or radiation. If so, what is mutation? Mutation refers to any change in the base sequence and or the structure of the DNA is known as mutation. And the substance that trigger mutation is known as mutagen. This mutagen cause what? Normal cell to become cancerous cells, or cancer cell, or known as carcinogen. So the mutation does not always give rise to the mutant's phenotype, and the most mutations are neutral or silent and have no effect on our body. Therefore, every mutation cannot cause what? The disease, or some mutation may be what? Neutral or silent. So only mutation in a cell that becomes sperm or egg cell are passed passed on to future generation. This means that only on the meiosis mutation can be associated with inherited inheritable, that it means. So mutation in other body cells only cause trouble when they cause cancer or related disease, and some cancer are raised because of the mutation in p53. This p53 can be very important to trigger the different cancerous cell or proliferation of the cell, and if the mutation can be associated in this p53, it can lead to different cancer disease. So this mutation can be divided into two, spontaneous mutation and an induced mutation are there. Spontaneous mutation means if it occurs in nature without any action of non-mutagen, and the induced mutagenesis or mutation is if a mutagen is used, or if it can be caused by the different mutagen, that is known as an induced mutagenesis. So this cause of mutation can be either spontaneous or induced. Spontaneous means replication associated with replication error, replication failures, oxidation, base losses, deamination or depurination, and adduction. Whereas induced mutation can be associated with replication error, replication failure, chemicals, and radiation like UV and X-ray or gamma radiation. Have a look and note. The most common UV-induced product is pyrimidine dimer, and the helix distortion that results from the dimer formation stops what? The DNA synthesis. So this DNA mutation can be divided into 5 categories depending upon the number of nucleotides involved or the type of defect associated with the mutation. The first mutation is known as point mutation or single base alteration. Here, what as the name indicates, includes what the base or the base substitution. Most frequently, insertion or deletion can be associated with point mutation. So a transition and a transversion are there here. 2 points are there. Transition means simply the replacement of one purine by another or that of one pyrimidine by another, but transversion is the replacement of one purine by another one pyrimidine and that of pyrimidine by the purine. So you have to identify these two terms. Transition, simply the replacement of one purine by another or one pyrimidine by another, but in contrast, transversion is the replacement of one purine by pyrimidine or pyrimidine by purine. And this point mutation can be caused due to different factors. One factor of point mutation is deamination. Adenine deaminate into hypoxanthine that base pairs with C and cytosine, and the cytosine deaminate into uracil that base pair with adenine. You know that cytosine is always paired with guanosine, but in case of deamination, this cytosine can be base paired with adenine, and the thiamine can be again deaminate and paired with guanosine, that it means. This is how nucleotide deamination can be takes place. Cytosine can be, you know that cytosine can be exactly paired with uracil if this amine functional group can be removed, or deamination means the removal of amine functional group in the form of ammonia. So this shows that how deamination can be takes place. Guanine may be changed into xanthine, and adenine can be changed to maybe hypoxanthine, and 5-methyl cytosine can be changed maybe thiamine. So this one is how the deamination can be what takes place. Mutagen or deaminating agent includes like nitrous acid and sodium bisulfite. These are the mutagen which can be cause or deamination or deaminating agent there. The second cause is depurination or depyrimidation. Here, the N-glycosidic bond is cleaved, leaving the unexposed deoxyribose attached to the sugar phosphate backbone, and the spontaneous depurination occurs at the 37 degrees Celsius at the rate of 5,000 to 10,000 cell division per day, due to what? Thermal liability of the purine to N-glycoside bond, spontaneous hydrolytic reaction, and the carcinogens such as mycotoxin and aflatoxin can be caused by this depurination and the depyrimidation. This also another cause of point mutation, so which lead to a different disease associated with mutagen. Here is the mutation brought by deamination and the depurination, and this shows that how mutation can be associated with change of the base pairing rule. In here the Watson-Crick base pairing rule can be broken due to the mutation associated with deamination and depurination. The third one is alkylation and adductive. If affects mainly guanine through the covalent addition of an alkyl radical, especially methyl, and the methyl halide, oxidative damage, ethylene methane sulfonate, and dimethyl nitrosamine are an alkylating agent which associated with alkylation and adduct formation so as to produce what deamination, so as to produce what mutation. You know that smoking cigarette contain a carcinogen, which is a mutagen that it means, such as aromatic polycyclic hydrocarbon benzopyrene. This benzopyrene can be what having the ability of what mutate the p53. And when this is compound is oxidized by cellular enzyme, it becomes capable of forming bulky adduct with guanosine. The modification of guanine residue interfere with hydrogen bonding in guanosine cytosine base pairs, producing distortion of the bond, which is a triple bond up. So both compound like aflatoxin and the benzopyrene can mutate what the p53 tumor suppressor gene that found in our body and are known the risk factor for liver and the lung cancer respectively. Therefore, smoking cigarette is highly prohibited because it can affect what this p53 tumor suppressor gene so as to lead us what the liver cirrhosis and also what liver cancer as well as lung cancers. This one is the mechanism of action, how benzopyrene can be associated with changing the base pairing or causing the oxidation in our body so as to lead to the cancer disease. So oxidation of benzopyrene and the covalent binding to DNA, this benzopyrene is not carcinogenic until it is oxidized within a cell, and then it is covalently bind to the guanine residue in the DNA, which can be disrupting the base pair or the bond of guanosine cytosine hydrogen bond of guanosine hydrogen base pair and producing the distortion of the helixes, which can be associated with proliferation of the cell or cancerous cells. So this all can show how alkylation can be takes place, and the alkylation guanine by ethyl methane sulfonate or EMS, which is an alkylating agent, can also cause another point mutation. There is also oxidative conversion of dioxyguanosine into 8-hydroxydioxyguanosine that rearrange into 8-oxodioxyguanosine that base pair with adenine, leading to GC to 80 conversion. This oxidation can convert to GC to 80 base pairing. So the base pairing can be converted in this oxidation. And the insertion or deletion of nucleotide also another cause of point mutation. Here a flat intercalating mutagen example, etidine, bromide and acridine intercalate between the base pair and the cause insertion or deletion of mutation that lead to the frame shift mutation. Especially this frame shift mutation can be associated with acridine, which can intercalate the base pair in the DNA structure. The other one is incorporation of base analog, for example 5-bromouracil, a thiamine analog, and the 2-aminopurine. This also another cause of point mutation. They are more likely to be incorporated during replication than normal nucleotide, and they induce 80 to GC conversion. This one is actually the reverse of oxidation. In both cases, they are the cause of point mutation there. Here point mutation have 4 effects or effects as follow. One, the point mutation can be produced or created in our body. It can have these effects. The first effect is known as silent or synonymous mutation. In case of synonymous mutation, the new codon specifying the same amino acid as the original codon due to what? The degeneracy of the genetic code here. So this silent mutation or synonymous mutation is associated with the new codon formation to the same amino acid to that of the original codon due to what? Degeneracy of the genetic codes. So the change lays in the third base of the codon, which differs among codons synonymous for the same amino acid that means. So it will not cause actually a functional change in the genome, and but it can change what create what the synonymous change in codon and anticodon amino acid or the DNA. Missense or non-synonymous mutation also there, in which the change occurs either in the first or second base of the codon, producing different amino acid than the original one. Especially sickle cell anemia or hemoglobinase and methemoglobinemia can be highly associated with this point mutation, especially due to missense or nonsense mutation. This nonsense mutation, in which the altered base results in the premature nonsense or termination codon, and this lead to what? A premature stopping of protein synthesis. Yeah. There is also read-through mutation, which converts the stop codon into an amino acid specifying codon that extends the produced polypeptide with additional number of amino acid, and this frame shift mutation, which reading frame shift in the trinucleotide code, can be associated. And actually this one is due to the deletion or insertion of the number of nucleotide not divisible by 3. Yeah, this one is. So any mutations that can be associated with deletion or insertion of a number of nucleotides that not to be divisible by 3 is known as frame shift mutation. Here the same reading frame shift occurs during translation and produce an abnormal protein. Example, beta thalassemia can be a disease associated with this frame shift mutation. Therefore, these 4 are the fate or the effects of what? Point mutation. So this one is a summary how this frame shift mutation can be delete or insert the unnecessary or codon in the starting, and the deletion and the insertion can be associated with this frame shift mutation. So the second type of mutation is 2-base alteration, that is due to UV-induced thiamine dimer, or this is due to thiamine dimer that induce the latter deletion mutation. So the 2 nucleotide cross-link in the same DNA strand caused by bifunctional alkylating agent. That means the third type of mutation is strand breakage. Here, single strand break are less harmful than double strand break in the phosphodiester bond, and the strand break are generally caused by ionizing radiation that has direct and indirect damaging effects through the free radical, radioactive disintegration of incorporated element, oxidative free radical generation reaction, and the chemicals, DNA strand breaking, chemotherapeutic agent like cisplatin, which is an anti-cancer drug. Especially this cisplatin can be what? A drug that can be obtained through serendipity for the first time by from the scientists that's checking what the effect of platinum on the bacterial duplication. During that time, you have to get what this is platinum can be given for cancer in combination therapy, or in alone. Nowadays can be used for cancerous disease. So this shows how the UV radiation can be created dimers, especially the formation of pyrimidine dimer due to this UV radiation. That means, so this UV radiation also another cause of what's mutation. That means, especially strand breakage can be associated with UV radiation also. The cross-linkage also another mutation. So 2 bases in the same or opposite strand can be cross-linked. So this DNA cross-linkage to histone or other protein can be associate another cause of mutation. And the chromosomal structure or a number of abnormalities, this means due to the faulty segregation, recombination, or gross damage, can be lead for chromosomal structure or number of abnormality. These all are the type of mutation that can be associated in our DNA. So this one is a summary. The types of damage associated to DNA: 1, single strand base alteration or the point mutation. This due to depurination, deamination of cytosine to uracil, deamination of adenine to hypoxanthine, alkylation of base, insertion or deletion of nucleotide or frame shift mutation, base analog incorporation. These all are the cause of point mutation. 2 base alteration can be associated with UV light induced thiamine pyrimidine dimer and a bifunctional alkylating agent, which can be cross linkage, and the chain breakers, ionization, radiation, radioactive disintegration of backbone element, and the oxidative free radical can be also associated with this chain break and the cross linkage. Also another damage in DNA, which can be associated with between bases in the same or opposite strand between DNA and the protein molecule. Example, histone protein can be also the cause of this cross linkage type of mutation or DNA damage. So if we see the DNA mutation, how this DNA mutation can be repaired. So DNA is the only macromolecule that can be repaired. Have a look at nature. Nature can be favor us because it can give another solution of repairing our DNA. So the cell have a multiple pathway to repair this DNA using different enzyme. In a cell, in your cell, the mechanism used for repair of DNA have, of course, many similarity have. But first, the distortion in the DNA helix is recognized, and the region containing the distortion is removed, and the gap can be maybe filled by the new template, and the gap in the damaged strand is replaced by the action of DNA polymerase that uses intact undamaged strand as a template. Finally, the ligase seals the nick in the strand that has undergone a repair. So ligase can be used for what? Repairing or for joining the two strands together so as to create what? The double helix DNA. So excision is one mechanism of repair. So this excision acts to correct many types of DNA damage affecting only one strand by replacing the damaged site by new synthesis. This excision repair can be associated with for a repair of DNA associated with what's point mutation. That it means it utilizes three mechanisms. One is base excision repair or BER. Here, it repairs a minor damaged nucleotide induced spontaneously or by base analog incorporation, alkylation, or bioxidation due to what? Ionizing radiation in the mitochondria and the nucleus, initiated by the cleavage of beta N-glycoside bond between the damaged base and the sugar, catalyzed by DNA N-glycosylase, leaving a pyrimidine or a purinic site. Next, this AP site endonuclease cleaves the sugar phosphate strand at this site, and then the DNA polymerase 1 or beta in mammals, polymerase beta in mammals and delta in yeast, insert the proper nucleotide dictated by the base on the damaged complementary strands, and finally the deoxyphosphate backbone is rejoined by the ligase enzyme, and the depurination are directly repaired beginning with epicytes endonuclease. That means. So this one is how excision repair can be, a base excision repair can be takes place. So this hydrogen bond base are there. In this hydrogen base, uracil and glycosylase can be inserted, and the gap can be filled. The DNA helix with missing base, so that base can be filled. Then after the base can be filled, the DNA ligase can be used for what? Joining the single strand so as to create what? A double strand DNA. The second one is NER or nucleotide excision repair. Here, although it is a backup system for the base excision repair, it differs from the base excision repair in that it is only nuclear, having a broader specificity, and it does not require initial base removal. Have the ability to deal with more extreme form of damage such as intra-strand crosslink and bulky base adduct. So a longer stretch of DNA, including the damages exist, and as a mechanism replace the photoreactivation system for the repair of pyrimidine dimer in organism devoid of what? Photoreactivation system, example in humans. So this NER or nucleoside excision repair can be associated with point mutation that caused due to what? Alkylation and adduct, as well as interstrand cross linkage. It also repair a strand break, interstrand base pair cross link, or DNA protein cross link, and a base modification example is the smoking induced benzopyrene guanine adducts, and it repair DNA strand damage of 2 to 30 base in length, and the first endonuclease cleave the abnormal chain and removes the distorted region. DNA helicase peels away a single strand containing the lesion. Now the DNA polymerase 1 delta or epsilon in human fill the gap with a new nucleoside using the healthy strand as a template. Then after, the DNA ligase can be the same fashion used for what? Joining the two ends of the DNA so as to create what? The original DNA. So this one is the diagram show that how the nucleoside excision repair can be takes place. Almost the mechanism is similar with the first one, so you have to see this diagram. And Here, xeroderma pigmentosum are there XP. This is an autosomal recessive disorder or disease due to what defective nucleoside excision repair caused by the mutation of at least 7 genes called what XPG with helicase, kinase recognition and excision activity. Therefore, if near or nucleoside excision repair cannot be performed this function, it can be associated with this disease, xeroderma pigmentosum disease. So the first defect to be identified was the defect of inexcision repair of thiamine dimers, most frequently due to the absence of UV-specific exonuclease, an enzyme that helps to remove what thiamine dimers that means. So patients are highly sensitive to sunlight or UV. This one is a symptom of xeroderma pigmentosum. And their skin is damaged and it is liable for multiple cancer, immunodeficiency, atrophy, and scarring of the eyelids, ulcerated cornea, or progressive neurological degeneration, and may lead to what a premature death also. So this one is the disease of xeroderma pigmentosum associated with NER. Have a look this. This one is xeroderma. Actually, xeroderma is a Greek word which means dry skin. That it means. So patient like UV endonuclease or exonuclease activity. So pyrimidine dimer due to exposure to UV light are not removed. So the clinical feature or symptom of this disease can be extreme sensitivity to UV light, excessive skin dryness, hyperkeratosis or thickened precancerous outgrowth of the dermis, blisters on the skin, corneal cloudiness or ulceration, hyperpigmentation, and finally atrophy of the skin can be what the critical features or symptom of this xeroderma pigmentosum. The third repair is mismatch repair, MMR mismatch based on the Watson-Crick base pairing rule are recognized by the enzyme of mismatch pair. So sometimes this Watson-Crick base pairing rule can be broken due to mutation, that this mutation can be associated with mismatch repair, and this enzyme determine which base of the mispair to be correct. So the correction can be taken by using this technique. So in bacteria, parental DNA strand contain a methyl group on adenine base or by dimethylase in a specific sequence of 5 guanosine, adenosine, thymine, cytosine base pairing. So during the replication, the newly synthesized strands are not immediately methylated. Before methylation occurs, a group of protein involved in mismatch repair can be distinguished parental from newly synthesized strands, and after this dimethylase can identify at which position the Watson-Crick base pair can be faulted or misly paired. So since this also cause another mutation, so this mutation can be corrected. It is also another cause of point mutation actually. So in bacteria, these proteins are called MUT-S, MUT-L, and MUT-H, with 5 got C3 endonuclease plus 5 more protein in eukaryotics, and the system identified the parent DNA strand or old by its 5-methyl adenine in the 5' guts to 3 sequence, whereas the new strand is not methylated yet. So after locating the mismatch or unpaired DNA sequence in the outer unmethylated strand, the defect, this is done with the help of helicase activity and special exonuclease or exonuclease 1 hydrolyze DNA into free nucleotide from the nick in either direction to the few nucleotide after that identification of the defect. Therefore, this DNA polymerase refill the gap and the ligase enzyme join the two ends was to create what the original DNA strands. So MMR can only aid replication fidelity if repair is targeted to newly synthesized strands. So methylated old strand, unmethylated new strand can be this one is a phenomena or the mechanism of action of mismatch repair can be takes place. So the next one is double strand break repairs. More dangerous type of DNA damage occur when both strands of the double helix are broken, because of here if both strands of DNA can be break down, the replication can be inhibited because there is no template actually. Yes, this leave no intact template strand to enable accurate repair. So this double strand breakage is the most dangerous mutation. So these double strand break are usually lethal to the cell, and they can be caused by high energy radiation, strand breaking, alkylating chemotherapy, oxidative free radical, and gene rearrangement. So this double strand breakage cannot be corrected by the previously described strategy of excision, excising the damage on one strand and using the remaining strand as a template for replacing the gap, because here there is no a template. Therefore, we need another mechanism to solve this problem. Non-homologous independent repair is known as a dirty solution for DSP. Although the mechanism is very error prone, it is often ineffective because the joining junction mostly involve non-coding sequence, and the defect in this repair system are associated with predisposition to cancer and immunodeficiency syndrome. Therefore, rather than healing, it may cause different disease. So this solution is known as a dirty solution for double strand breakage. So the homologous recombination repair, this one is the much more accurate type of DSP repair that occurs in newly replicated DNA. Here you have to replace the original sequence using the homologous chromosome or sister chromatid as a template. So, occurs in S phase and the G2 phase of the cell cycle after a sister chromatid has been created. So this one is actually the correct and error prone mechanism of solving the DSP mutation. Here sister chromatid can be created, and this sister chromatid can be used as a template. In that template you have to repair, and you have to create solve the DSP or another strands of DNA, which can be again joined by using one ligase enzyme. So this one is all about the part 2, the part 3 of nucleic acid. The last part 4 may be continued after this slide. So thank you for watching and for your attention. So at the end, don't forget to subscribe the channel, comment the channel, like the channel, and share for your fellow friends so as to get this interesting tutorial video of medical biochemistry and the most important chapter 3, which is all about nucleic acid.